Wait for the others. So, I'd like to understand what's your thought on the environmental monitoring, which we are going to continue discussing today. What's your thought on it? Any thoughts? Any thoughts on it? Any idea?
Monitoring the environment to ensure that ensuring that it does not pollute the environment before releasing any anything into the environment. It has to be make sure it is treated and then released.
So, what happens in case it's contaminated? So, what happens if in case there are some particulate matter and all that stuff? So, what happens? Any idea? What about the others? How come they are not talking?
It causes severity to the first people and area also area.
It affects the quality of the drug form.
Okay. Anybody else? And how do you think it is done?
If there is a particulate, we have to, if there is a contamination, we have to inform to the supervisor and we have to do the investigation, and kappa we will be doing corrective action and preventive action.
Okay. What about the viable counts, the non-viable counts? How are those handled? So you completed the session on environmental monitoring and it's all gone way above and beyond, is it?
Viable particle count.
I can't hear you. Sorry, please.
Hello? Hello?
Can't
Hello?
hear you. It's very feeble.
Ma'am, non-viable particle can be controlled through the HVAC by controlling um, the airflow. The particles can be reduced.
Okay.
We'll do that active air sampling and passive air sampling, ma'am, to see if there is any particle contamination.
Okay. Fine. So now we move on to the stability aspects. Day before yesterday, we did speak about stability of of what did we speak? Stability of what did we speak on Tuesday? If you can jog my memory. Stability of API is something that we discussed. Yes.
ODS. About ODS.
What about ODS?
Oral dosage forms.
We spoke about the stability of the product. If you look at it, we did discuss what you mean by stability. Just I'm briefly going through. I'm not going to get into the details. What do you mean by stability study? Why is it so important? Here, I'd like to indicate that if you remember the journey of the molecule, okay, it goes through the journey of discovery, development, non-clinical, clinical, and manufacturing. It goes through the entire journey. In that, what is important is that the minute the molecule, you understand more about the molecule, the stability of the molecule gets sort of, evaluated, especially during the development phase, and that moves into the non-clinical and manufacturing phase. That's where the stability is very, very important. So until the stability of the product, of course, you know that generic products, that means which are already existing, and you're trying to have an equivalent product. So in those cases, what happens is that they would have some thoughts on that particular molecule. And what they will do is they will ensure that the retest period is evaluated in the beginning. Typically, in the beginning, they give retest period, and over a period of time, once they start getting the data on it, they will indicate it as the expiry period. Okay. During the stability studies, it's typically known as retest period, especially in the early stability studies, early development stability studies, so to speak. Especially in clinical trials and all that, what they do is, I have seen cases where the drug will have a retest period of 8 months. And as the 8-month retest is coming closer, they would have evaluated the drug again, and again they will extend the retest period. So like that, they may extend it a couple of times till they get the stability points. One is that, and secondly, by then the molecule would have gone into the manufacturing stage, commercial manufacturing stage, by which, again, we discussed the other day that 3 batches needs to be put in over there for stability. Okay, and that will be sort of giving the idea as to what is the status of the stability. So retest period is what they give during the early stage of the development of the molecule. And that's why you may see in some cases, especially development of molecule, retest period is indicated. I'm not talking of retest of raw material. Here I'm talking about retest of API and retest of the finished drug form. Okay, that is drug substance and drug product form, not the raw material. Okay, please bear that in mind. Raw material retesting again is dependent on the manufacturer. Okay. So let's get into the actual slides here. And again, stability studies are done, and of course, they do it in these phases. This is what I just briefly explained now. They were especially the environmental factors related to temperature, humidity, and light, and also the packing material. We did discuss about the primary, secondary, and tertiary packaging. And again, it enables the packaging of the materials. So if you notice, probably you may have taken crocin syrup, when you were a child, and now you're taking crocin tablet. So you have seen the kind of packaging that has evolved. Earlier, it used to be glass. Okay, but now what we are seeing is material that is inert, and it is probably silicone material. So again, there are variety of packaging material that has come. Packaging material itself is a huge industry. And of course, there's a lot of opportunity for people like you and me. Okay. Here again, it is to establish retest period of the drug substance or a shelf life for the drug product and recommended storage conditions. So what, if you look at the period, it is always accompanied by the temperature. And if it is light sensitive, they also say not in direct contact with light. Okay, and of course, they would have indicated the temperature. They would have, if in case it is 2 to 8 degree, then they would have indicated the temperature. So the retest period or the shelf life of the drug product is always along with that of the temperature. Please remember that. Okay, so the scope, the rationale, the advantages, it's a legal requirement more than anything else. And of course, patient safety always comes on top because, as I said earlier, that we can't have a drug that is sort of, reduced in its efficacy within the stability period. So that's the reason why in the DCGI office, that is our drug controller office, there is a testing lab dedicated for testing of material. So what they do is they do take samples from the pharmacist, the pharmacy. They do take samples, they'll take it back to their lab and they do test it. Basically to verify whether whatever we claim is what is being supplied to the pharmacy and to later to the patient. So again, chemical stability, physical, microbiological, therapeutic, and toxicology. So now what happens is in case of chemical or more than chemical, physical, sometimes over a period of time, denaturation occurs. It looks very discolored, rather, so to speak. It would have discolored. It would have been initially white color product, but over a period of time in the stability study where they would have kept the samples, it would have denatured or it would have lost its appearance. It may have in the beginning, it may have a good finish, shiny finish, but however, it may have become dull or it would have absorbed moisture, it would start disintegrating. That means it would start becoming brittle and powdery. So all these things are very critical. The way the stability study is done is very, very critical. Of course, there's microbiological, especially when it's related to sterile products. And of course, the microbial growth also they verify whether there are any small particulate matter inside the, the solution, all that would be reviewed. And again, in stability studies, there are different stages, 1, 2, 3, 4, 5, and 6, of course. But again, this is where the, integrity of the molecule is checked. That is, stress and accelerated testing is done. Again, they do pre-formulation batches. And then they do on scale-up batches. Again, this is all in development labs that will be done. There are special formulation development labs, FDP, process development plan, or it would be even in analytical also. Accelerated and long-term testing for registration purpose. Again, if it's a legal requirement, and ongoing stability studies, and of course, follow-up stability studies. During the stability testing, the development or the characterization of the compatibility of the material will be verified, stability of the API will be verified, and early formulations will be verified. Factors are temperature, light, moisture, etc. So again, if there are any issues with the cover of the container, as I said, extractable and leachables, that can have an impact on the stability of the drug. And also exposure of the drug formulation to the oxygen also affects the stability. we did look at the ICH guidelines that are available. Predominantly across the globe, people use ICH guidelines. Again, we did discuss about the zones, and we do come under Zone 3 and Zone 4 countries. Okay. The active pharmaceutical ingredient. These are the various aspects that were reviewed. Container closure system. So what happens is container closure system plays a very, very important role. Any crack in the vial or any crack in the bottle or a tear in the, what do you call that? Blister packs, that is the tablet, what we see, can impact the quality of the drug. Now, if you look at the blister packs, if you next time, whenever you get a chance, I want you to just have a look at the layering of the blister pack. There is an aluminum foil on top. Then there is a, what do you call, a layer of, again, HDP kind of a material, thin layer of that. And then you have a foil again coming in. If you notice the product logo, the product details are on the top of the blister pack, not inside. Inside, it is just plain foil. Okay, so that again, they would have done stability testing and again, extractable, leachable information, based on which they would have decided that, yes, this is non-reactive and hence it can be used. So it is actually in a couple of layers. This is the, again, storage conditions, RH 2560, 3065, 3065, 4075. So this is how it's typically declared. Drug substance, again, in refrigerator and freezer has been indicated here. Stability commitment is there. So this stability commitment is given during the dossier submission to the regulatory authorities. So if you notice, when the data submitted is from fewer than 3 production batches, a commitment should be made to continue the long term with additional production batches to a total of at least 3 batches. So we move on to that of the drug product. That is the product that you and I consume. So again, if you notice, general photostability test, selection of batches, container closure systems, specifications, testing frequency, storage conditions, stability commitment, evaluation, and statements and labeling. So typically, again, the storage statement should be established for the labeling based on the stability of the drug substance. That means, based on the drug substance, you would be indicating that of the drug product as well. That will be the basis for your stability study for drug product. Like, for example, if your drug substance is a statin, or if it's what do you say, insulin, then the stability of the drug substance that you would have done will be the basis for the drug product. So it should be based on the knowledge and behavior of it from the stability studies and on experience gained in the clinical formulation studies. So in terms of photostability studies, it is exposed drug product outside the immediate pack, in the immediate pack, and in the marketing pack. That means in the final carton as well. So there are 2 different options it's given. Artificial day lamp combining both visible and UV, similar to the requirements of D65 and ID65. Again, this is a requirement as per regulatory. And again, it's option 2 is white fluorescent and UV lamp. again, as per the requirements of the ISO standard. So please understand that whenever there is a claim test, what do you mean by claim test? For example, if you're talking of a claim test like, say you're using Dettol to wash your hands. And, there is a claim made by the company that says that it will remove it will remove all the bacteria from your hands, or some portion of the what do you call contaminant is removed. Or for that matter, if you look at the case of hand sanitizer, there are certain claims that are done. Or zero germ what do you call disinfectant solution. So when you see those, please remember, it's not just a claim that they're doing for marketing. It's much more than that. It is something known as a claim test that is done, and it follows certain ISO norms. There are standards for claim tests, which the industry follows before they put these claims on the bottles that you and I use. Same thing also in case of stability. There is a guidance for stability, which is done, and based on that, expiry is derived, and based on which we consume those medicines within the expiry period. Now, coming to selection of batches, it should be again as per 3 primary batches. It should be of the same formulation and packaged in container closure system as proposed for marketing. Like I think I have the thyroid medicine here. And this, I don't know if you can see, this particular bottle itself will be kept for stability. It's not that they will remove the packet and transfer it into a smaller, what do you call, self-sealing cover or Ziploc cover and keep it. Okay, so this is something that you need to keep in mind, that this product itself is stored in stability. Okay, and 2 of the 3 batches should be at least pilot scale batches, and the third one can be smaller. It should be performed on each individual strength and container size of the drug product unless bracketing or matrixing is applied. So now what happens is, Thyronorm, the thyroid tablet, comes in various dosage forms. This is 25. Okay, then we have one for 50, then there is one for 62.5, then there's one for 120 and all that. So you have to do the stability for all the strengths. That's the requirement, because you never know if it can vary. Container, it should be conducted on the dosage form packaged in the container. That's what I just said. It should be conducted on the dosage form packed, proposed for marketing. It should cover the physical, chemical, biological, microbiological attributes and the preservative content. And it should be fully validated and stability indicated. So here I'd like to share an incident that happened when I was heading the quality for clinical trials. It was a major rheumatoid arthritis study involving nearly 2,000 patients across the world. 2,000 patients across the world, many countries were involved. And the rheumatoid arthritis, the subjects were all menopausal women. That means the women were 60 plus, most predominantly 70 plus, and they were, part of the study. So what happened was the subjects, the patients were given the calcium tablets. So the drug was different, but the drug was a rheumatoid arthritic drug. Okay, but however, as a supplement, the calcium also was given to them. So these women had taken the calcium bottles and they had taken it home. And I think somewhere in Mumbai, they came back saying that the bottles were stored in this. Tablets were stored in this rather. And they came back saying that the tablets had black dots on it. Very, very minute blackish grayish dots. very minuscule to the eye, but they still could find it. And they came back to the site, to the doctor, and they told them that, see, this is what the medicine is showing. And the doctor in turn alerted the pharmacist and to the sponsor and to the CRO. And it was an MNC company, Merck, so to speak. So they came over and they audited us extensively. They said, we ship it to you, and you in turn ship it to the hospital. Hospital in turn gives it to the patient. So they wanted to see in the chain of custody if any issues were there. So they come and did extensive audit on us. About 4 days audit they did, seeing how we packed it in the cartons to different sites, etc., all that stuff. Then finally they went to the site and they saw that that complaint had not come from any other site. That complaint had come only from that site and only from that particular patient, 2 or 3 of them. So then they concluded that it could be that they had not stored it properly. They may not have had proper storage conditions at their residence. So you never know if the drug was stored properly. They may have stored it near the heat source. They may have stored it near the water source. No, I know, I agree that the bottle should be completely integral. It should not allow external influences happen, but we don't know what had happened. Okay, so it was inconclusive. That means definitely not the manufacturing site, not where we stored it, not the doctor also. It was only at the patient site. So this is how they concluded. So again, testing frequency. 1 year every 3 months, the second year every 6 months. That means if it is 2 years, for the first year, it happens every 3 months, and then later on it happens for 6 months, and the subsequent years is annually. And again, T0 is the time 0, T final. It should be evaluated for its thermal stability and sensitivity to moisture or potential for solvent loss. And it should be sufficient to cover storage, shipment, subsequent use with due regard to climatic zone, and photo stability at least on one primary batch. So what they do is, if you notice, there's so many conditions on it, right? It can be very confusing. So what companies do is, what the QC lab does is, they prepare a stability protocol for each and every tablet, for each and every dose of the tablet. That means the different doses that are there. For each and every dose also, they have a separate stability protocol. Again, long term. $25, $60, $30, $65, $40, $75. And again, the months, the duration, these are considered. So when do they say there is a significant change? It is defined when 5% change in assay from its initial value, any degradation products exceeding its acceptance criteria, failure to meet the acceptance criteria, especially related to physical attributes, as I said, and like caking, and then the solution becomes thicker, it becomes like a slurry. All these things have to be noticed. Some changes may be expected rather, and again, depends on the dosage form. So coming to drug products packaged in impermeable containers, sensitivity to moisture or potential for solvent loss is not a concern for drug product packaged in impermeable containers. So studies can be conducted under controlled or ambient humidity condition. Coming to semi-permeable containers, aqueous-based products packaged in semi-permeable containers should be evaluated for potential water loss. Again, the requirements are indicated over here. The intermediate condition, again, there are initial 6 months, 9 months, and 12 months. So in case of storage in a refrigerator, there is long-term and accelerated storage that is done. And again, in freezer as well. These are, mind you, dedicated freezers and dedicated refrigerators. And 3 production batches, that's a commitment. And accelerated, that's for long-term. Then 6 months for accelerated. And if it's less, again, the same commitment should be there to continue the long term and to place additional production batches. And again, the analyst must find the batch to batch variability, and if it's small, only then it is accepted. Of course, they will be doing a statistical analysis on that. So coming to statements and labeling, storage statement should be there on the stability evaluation of the drug product and the drug substance. And instruction should be there. And ambient conditions and room temperature should be avoided. And retest period should be derived from the stability information, and the test date should be displayed on the container label if appropriate. So in clinical trials and all, if the product is under retest, they do put a sticker, a dedicated sticker for it. So coming to ongoing stability study, it is to monitor the API and finished product remains within the specifications. It should be described in a protocol, 1 production batch per year, tested annually. That's the requirement. So let's understand what causes the instability. One is in case of oral solutions, it is loss of flavor, change in taste, off flavor presence due to interaction with plastic bottle. This is what I said, leachables. Loss of dye. The dye may sort of, fade away. Precipitation, discoloration, change in smell, and use of a proper excipients and suitable packing material. So there was this case wherein the patients complained that the product had a very bad odor, fishy smell. Not fishy, that is the dead fish kind of a smell, and or dirty sock smell, foul sock smell. So this is what they complained. And when they investigated it, they found out that the container was leaching the chemical into the product. And that's the reason why the smell and the other issues were coming up. So coming to instability problems is again disintegration time. Some molecules take a longer time for disintegration. Okay, dissolution profile, hardness and appearance, change in drug release is the effect because it can impact the release of the drug. Design of the product based on proper pre-formulation studies will prevent from its instability. Now coming to the capsules, it is the instability problem is change in the appearance, dissolution and strength. And very specifically, it's on change in drug release. So again, for everything, the product needs to be designed in a very appropriate manner. Coming to emulsions, it occurs due to droplet diameter, viscosity, density, temperature, pH, presence of microbe, and loss of drying content uniformity in different doses. And again, you need to relook at the design of the product. Now, coming to suspensions instability product, it could be because of the particle diameter, concentration, viscosity, temperature, pH, microbe presence. It could be because of caking, crystal growth, or settling. So it's again the loss of drug content uniformity. Again, you need to relook at the formulation. So if you notice, this is how the company stores. So this is one of the stability chambers, so to speak, you can say. You notice each has a sort of a carton in it, plastic bin, and the product will be stored within that particular bin. These are the following scenarios: development phase, registration dossier, post-registration period, post-validation batches, key starting material, batch size, equipment, vendor, manufacturing formula, primary packaging material. And again, all these are indicated in the protocol. It contains the typical requirements of purpose, scope, responsibility, label claim, references, product information, list of tests, and once the analytical is done, you need to document it, and it shall be compared with the previous results of the previous batches. Any changes is indicated in the SOP, as per the SOP, in either OOT or OS. Stability trends will be determined in real time. Results will be evaluated. Summary report will be prepared, and it should contain the necessary information that's indicated here. What is hold time study? And it's basically to establish time period for which materials may be held under specified condition and will remain within the defined specifications. Again, these are the tests: assay related substance, microbial test, LOD, water, bulk or tap density. It is typically carried out on the first batch for newly introduced product. And it shall consist of different stations for analysis. QA department shall prepare the report and make recommendation about the manufacturing stage. Based on the hold time study, recommended hold time shall be mentioned in the respective batch records. Storage conditions shall be clearly labeled. Samples will be stored in the same pack as that of the storing in the intermediate. If hold time of intermediate exceeds from established hold time, intermediate shall be retested. Again, it shall be go through the deviation management process. Demonstrate as appropriate late exposure does not result in unacceptable changes known as photostability. The physical characteristics of the samples under test, the effects of changes in physical status, interactions between the samples. At the end of the exposure period, the sample should be examined for any changes and for assay. The force degradation studies provide suitable information to develop and validate test method for confirmatory studies. It should be capable of resolving and detecting photolytic detergents that appear, degradants that appear during the confirmatory study. Special labeling of packaging is done. While evaluating the results of photostability studies, it is important to consider the results obtained from other formal stability studies. In use is to provide information for labeling on the preparation, storage, condition, and utilization period of multi-dose products. And repeated opening and closing may pose a risk for the product quality. In use stability study, it should be set up to simulate the use of product in clinical practice. Suitable quantities of the product stored at the recommended use storage condition should be removed. The chosen sampling point times, withdrawal method and amount should be aligned with the product leaflet. That means whatever sample you're going to withdraw should be apportioned to the product quality requirements. Excuse me, can somebody read this, please?
Yes, ma'am.
Yeah.
The study in use stability study design, number of batches to be included. The study should be set up as far as possible to simulate the use of product A. Minimum of 2 batches, at least at pilot scale, should be included in the study. At least one batch should be tested near its end of shelf life. If at the end of shelf life, in-use stability data are not available at time of submission of the registration dossier, one batch should be in use tested at the final available time point before submission.
Next.
The appropriate physical, chemical, and microbial properties of the product susceptible to changes during use should be monitored. Physical, color, clarity, closure integrity, particulate matter, particle size, chemical, active substance assays, antimicrobial preservatives and antioxidants, degradation levels, pH, microbial total viable count, and sterility.
Next.
In-use information to be included in the registration dossier of a multi-dose product, tabulated in-use stability data with an assigned in-use shelf life and rated in-use storage condition, or a justification why an in-use shelf life is not required. Example, on the basis of experimental data, should be included in the registration document. In-use information to be included on the label of a multi-dose product. The applied in-use shelf life should be reported there. In addition, if feasible, a dedicated space should be present on the multi-dose product label to allow the user to record the data of first container opening or the in-use product expiry date.
So, dedicated space should be present on multi-dose product label to allow the user to record the date of first container opening or in-use product expiry date. Next, please.
Good preparation for an in-use study will ensure proper execution to achieve its goals. Items that should be in place prior to study execution are: the in-use study should follow a protocol that has been approved by management and quality. Validated test procedures should be in place. For example, the testing should include test for appearance or clarity, pH, assay for potency, assay for impurity, related substance, and particulate matter. Do not assume that every testing procedure can be performed as written. Methods for potency and impurities may need adjustments to sample preparation scheme or HPLC injection volume to account for the different sample concentrations. Knowledge of how the product has been used in the clinical trials or how the reference product has been used in a clinical setting is necessary. This information will determine syringes, IV bags, diluents, and transfer lines that will be needed. This information will also describe how the product is reconstituted, storage conditions prior to use and during use, and during use. What?
And what would be
I can't see
longest
that
time
line.
period
And what
prior
would be
to
the
use?
longest time prior to use?
Freeze-thaw study, very important one.
To determine if your formula will remain stable under various conditions, this type of test puts your sample through a series of extreme rapid temperature changes that it may encounter during normal shipping and handling processes. Freeze thaw stability testing is highly recommended, especially for liquid based cosmetics. These products may experience phase separation that can negatively affect the intended function. Freeze thaw testing is conducted by exposing the product to freezing temperatures, approximately 10 degrees Celsius for 24 hours, and then allowing it to thaw at room temperature for 24 hours. The sample is then placed in a higher temperature, approximately 45 degrees Celsius for 24 hours, and then placed at room temperature again for 24 hours. The sample is analyzed for significant changes. This completes one cycle. If after 3 cycles of freeze thaw testing, no significant changes are observed, you can be confident that the stability of your product is sufficient for transport.
Okay. This is especially for liquid-based products. Water loss study, please.
Aqueous-based products packaged in semi-permeable containers should be evaluated for the potential water loss, in addition to physical, chemical, biological, and microbiological stability. This evaluation can be carried out under condition of low relative humidity, as discussed below. Ultimately, it should be demonstrated that aqueous-based drug products stored in semi-permeable containers can withstand low relative humidity environment. If the FPP is packed in the semi-permeable container, appropriate 873 information should be provided to access the extent of water loss.
Again, because water loss can impact the quality of the product. Okay. Example of an approach for determining water loss. This is an example that has been quoted here. We can go into the detail later. Please remember the size of the container, the quality of the container. Till now, we spoke about the temperature, humidity, etc., water. But here, in this case, the container closure system, the container size, what quantity needs to be filled, everything is very well designed. Okay. Next, please. Shelf life.
Shelf life estimation details. An expiration date is defined as the time up to which the product will remain stable when stored under recommended storage conditions. Thus, an expiration date is the date beyond which is predicted that the product may no longer retain fitness for use. If the product is not stored in accordance with the manufacturer's instructions, then the product may be expected to degrade more rapidly. Shelf life is the time during which the product, if stored appropriately as per the manufacturer's instructions, will retain fitness for use greater than 90% of label claim of potency. The expiration date is also defined as the date placed on the container or labels of a drug product, designating the time during which a batch of the product is expected to remain within the approved shelf life specifications, if stored under defined conditions, and after which it should not be used.
Next. Next person.
The shelf life is determined from the data obtained from the long-term storage studies. The data is first linearized and test for the goodness of it is applied. The linearized data is then analyzed to see that the slope and the intercepts are matching. For determination of significance of difference in case of slope or intercept, statistical tests like t-test should be applied. The data is available in the form of only 5 data points, that is 0, 3, 6, 9, and 12 months, either pulled from the 3 batches or from the 3 individual batches if they are not fit for pooling. In case data is not fit for pooling, stability estimates are to be made on the worst batch. The shelf life or expiry date is determined from the regression line of this 5-point data based on the calculation of 95% one-sided confidence limit.
For reading the expiry date, 90% during concentration is considered at the lowest specification limit, and the point where the extinction line cuts the 95% confidence limit line is taken as an expire date. For new drugs, it is generally practiced to grant only 2 years expire instantly, which is based on the satisfactory 1 year long term and 6 months accelerations stability data. The expiry date for third and last years is allowed only on production of real-time data for subsequent years. Most pharmaceutical products are characterized by only one shelf line. However, in some cases, a product may be have two. Examples: a freezer dried Lyophilized protein product may have only one shelf life, say 2 years for the product stored in the dry conditions, and a second shelf life, say 2 days for the product when it has been reconsidered when appropriate vehicle and is ready for injection.
Bracketing and matrixing. Bracketing, the design of a stability schedule such that only samples on the extremes of certain design factors, example strength, container size, and or fill, are tested at all time points as in a full design. Design factors are variables to be evaluated in a study design for their effect on product stability, strength, container closure sizes, and or fills.
Strengths applied for multiple strengths and clauses related formulation. Examples: capsules, different fill, plug size, and strengths made up of same powder blend. Tablets, different strengths manufactured with compressing varying amount of same granulation. Oral solution, different strengths which may differ in minor excipients, colorants, and flavoring. If different excipients are used among strengths, bracketing should not be applied. Container size or fills. Either fill size or the container size should be vary and other should be constant. If both are varying, the smallest and the largest are not considered as the extreme of the packaging operations. In selecting the extremes, compare the characters carefully because it may affect the stability of the product. The characters includes containers walls, thickness, surface to volume ratio, and oxygen permeation rate per dosage unit, closure geometry. It is applied for the same container having a different closures with justification.
So basically, these are, you could say, guidelines, like when they are having similar kind of strength, different strength product and similar size of the tablet. So there are a lot of permutation and combinations, and these are well thought of when they are deciding to do the stability studies. Because if you have the same strength and they have the same what do you call pack size or different strength, again with slight variations in the excipients. So based on this, they will go by the requirements of the particular drug substance and see how it would have behaved during the development and then take a call. Now coming to matrixing, it is the stability schedule in which the selected samples are tested at a time point. At subsequent time point, another set of samples of all combinations are tested. Again, this is indicated in the stability protocol. So whatever I'm saying is very explicitly written in the stability protocol. The stability protocol can run into probably nearly hundreds of pages as well, depends. And again, the differences in the same drug sample at different strength, different batches, different sizes of the same container closure system is also considered. The design assumes that the stability of each set of sample represents the stability of the remaining samples at a given point. That means including these different strength and batches. When secondary packaging system contributes to the stability of the system, that also needs to be tested, especially the packaging material. So these are designs, matrixing designs. Different strengths. And following should be considered, that is knowledge of data variability, the stability of the product, availability of supporting data, difference of the product within a factor or number of factors combinations in the study. Some of the deficiencies that we notice is the size of the lots used in the trial, clearly packaging material, accumulate stability data, define accurately the temperature and humidity conditions, fully describe test methods. So what happens is you have the stability protocol. Okay, in that protocol, the test methods will be indicated. That's the reason why it goes into many pages. Test method is indicated or test method is referenced. And of course, then they have a table that indicates, what stability time point, what is the pull-out sample, etc., etc. What is the label that needs to be stuck on? Then what are the, you could say, observation data sheets are also included in that. That means each and every stage of the stability, there are observation data sheets that are part of the stability that comes in as a part of the stability study plan. And again, how are the out of specifications, out of 10 results done? How should you extrapolate the data? That may not be indicated. Failure to provide results from various stations to facilitate assessment of any trends. Failure to include information on the physical characteristics of the product. Failure to conduct additional tests or investigate. Sometimes there could be a data integrity issue as well. So how is that handled? All these are also taken into consideration. So the responsibilities, of course, R&D, then the QA, then of course the various other QC teams, they are responsible, and regulatory affairs also, because they are the ones who review the stability protocol as well. The quality head would approve the stability document. These are the definitions which you would be looking at in the standard. Any questions? Okay, it's about 10:05 now, so we come back by 10:20.
Yes, ma'am.
10:25 is also fine.
Yes, ma'am. Thank you, ma'am.
Is everybody in?
Yes, ma'am.
So what I want you to do is to, this is going to be a assignment which you as a team are going to work on it. Okay, so I want you to go to the website ich.org. Even I am opened it. Okay, and I want your team, your entire group to be split into 3. Okay, and I want one team to work on the stability data package for registration application in climatic zones 3 and 4, which we belong. I just want you to briefly tell me what are the critical points to be kept in mind. Okay, that's one team is going to work. I'm again not going to split. You have to decide which team you want to belong to. 3 stability teams. One is related to this. The second one is related to bracketing and matrixing design. Okay, take one drug product, not a drug substance. Take one drug product and just indicate how would you plan out for a sterile product. Okay, and the other one is the last one. The third team is going to work on new dosage forms. New dosage forms in consideration with the Q and B also. You need to consider, of course, it would have considered, but just want you to let me know on Q and B as well. So it's going to be 3 teams. One will work on Q1F. The other one will work on Q1D. The other one will work on Q1C in relation also with Q1B. Okay. And I am giving you about an hour's time, including the break for the next session. That's between 11:15 to 11:30. So you have about 45 minutes now, 10:30 to 11:15. Yeah, and a break in between as well. So we meet at 11:30, please. Is that okay? Yeah, thanks.
Yes.
Again, I'm not going to split you, but
pack and then in the marketing pack. For some products where it has been demonstrable that the immediate pack is completely impenetrable to light, such as aluminum tubes and cans, testing should normally only the conduct on directly exposed drug product. It may be appropriate to test certain products such as infusion liquids, thermal creams, etc. to support their photo stability is in use. The analytical procedures used should be suitably validated. Presentation of samples. Whether practicable when testing samples of the drug product outside of the primary package, this should be presented in a way similar to conditions mentioned for the drug substance. The samples should be points nice to be provide maximum area of exposure to the light source of the example tables, tablets, capsules, etc. should be spread in a single layer. If direct expose is not particle, example due to oxidation of a product, the sample should be placed in a suitable protective inner transparent container. Analysis of samples. When powder samples are involved, sampling should ensure that the representative upturn used in individual test for the solid oral dose from products. Testing should be conducted on the appropriately sized composite for example, 20 tablets of capsules. Similar sampling considered such as homogenization or solubilization of the entire sample applied to other materials that may not be homogeneous for exports. Judgment of results. Depending on the extent of changes, a special labeling or packing may be needed to mitigate exports or to light. When evaluating the result of photostability studies to determine whether change due to exposure to light is acceptable, it is important to consider the results obtained from other formal stability studies in order to ensure that the product will be within proposed specifications during the shelf life. That's about team 1, ma'am.
can you tell me the title of the standards that you referred to? The ICH Q1, which standard did you refer to?
QB and QC, ma'am.
QC and D. Okay. The next team, please.
Good morning, ma'am. I'm Prashanti. We are team 3. So our topic is stability data package for registration applications in climatic zones 3 and 4. So first, I'll go with the introduction. The purpose of stability testing is to provide evidence of how the quality of an API or finished pharmaceutical product varies with time under the influence of a variety of environmental factors such as temperature, humidity, and light. The stability testing program also includes the study of product-related factors that influence its quality. For example, like interaction of the API with excipients, container closure systems, and packaging materials. In fixed dose combination, finished pharmaceutical products, fixed dose combinations, FDCs.
Sorry to interrupt. Sorry to. If you have any material, can you show it on the screen?
For my part, there is no presentation, ma'am. For another person, it is there.
Okay.
So the interaction between 2 or more APIs also has to be considered. As a result of stability testing, a retest period for the API in exceptional cases, for example, for unstable APIs, a shelf life is given, or a shelf life for the finished pharmaceutical product can be established and storage conditions can be recommended. An API can be considered unstable under the conditions studied in a particular type of packaging when a significant change is observed. So various analyses have been done to identify suitable testing conditions. So the members of WHO stated that have notified WHO of the long-term stability testing conditions they required when requesting market authorization are listed in long-term stability testing conditions as identified by WHO member states. So now we will see how APIs and finished pharmaceutical products will undergo the stability testing. Thank you, ma'am.
So for the stability testing of API, the shelf life assigned for the API by the API manufacturer should be derived from the stability testing data. For that, stress testing may be carried out on a single batch of API. It should include the effect of temperature in the 10 degrees Celsius increments, for example, 50 degrees Celsius or 60 degrees Celsius above the temperature used for the accelerated testing. Humidity, for example, 75% relative humidity, RH, or greater, where the appropriate oxidation and the photolysis of the API. The conditions studied should cause the degradation to occur the small extent, typically 10 to 30% loss of API as determined by assay when compared with non-degraded API. Results from these studies will form an integral part of the information provided to the regulatory authorities. After that, the selection of batches will be done. For that, the data from the stability studies on at least 3 primary batches of API should normally be provided. The batches should be manufactured at a minimum of pilot scale by the same synthesis route as the production batches. After that, container closure system. The stability testing should be conducted on the API package in the container closure system that is same as or the stimulates the packaging proposed for the storage or distribution. The stability studies should include the testing of stability indicating attributes of the API, that is, those that are susceptible to change during the storage and are likely to influence the quality such as safety and efficacy. The testing should cover as appropriate as physical, chemical, biological, and microbiological attributes. Now Joshua will continue.
Good morning, ma'am. I'd like to talk about the um, testing frequency. For the stability of the API, it has to undergo the you have to undergo the long-term stability test. For that, the frequency of the test also matter, and for a for long-term stability, for 1 year, every 3 months it is studied, and for 2 years, every 6 months, and for then I'll talk about the storage condition. The storage condition of the API is studied whether it has the thermal stability and sensitivity to moisture, and it is monitored and controlled and recorded. And for new API for long-term testing, minimum 12 months it is studied, and for existing API, a minimum of 6 months is studied, and we have the long-term as well as intermediate um, um, table for that. I like to show. Um,
Are you the three people who are presenting? Sharing a screen. Okay.
yes, we have the long-term and the intermediate study. From the long-term, the storage condition maybe 25, 30, and 30. And for it is maybe for the 12 months or 6 months, and for intermediate, 30 degrees for 65% of RH for 6 months. Similarly, we have the study for API in the refrigerator in long term as well as in accelerator study. Long term for 5 degree for the 5 degree Celsius plus or minus 3 degree for a minimum of 12 months or 6 months for new existing one. And accelerated, we have three storage condition 25, 30, and for 6 months. And similarly, API is also studied in the freezing condition for a long term of minus 20 degrees Celsius to plus 5 degrees Celsius for 12 months or 6 months for 6 months. Then for evaluation of the it has the written protocol and the result. If there is a minimum of 3 batch of the API is taken, then if there is any degradation, um, if there is a degradation in the stability test, either retest is granted as then in the statement and labeling, it have they have to display the stability condition of the API, and terms such as ambient conditions or the room temperature should be avoided. For ongoing stability studies, it is a continuous monitoring so that it meets to ensure that it has a within its specification, and for ongoing from a for stability monitoring program, for it is taken for single batch, um, for new sorry um, it is it is a continuous monitoring so that it is a it meets up the specification, and that's all from team Premium.
Good afternoon, ma'am. My name is Sanina, and I will be talking on the stability conditions of finished pharmaceutical products. First, we will do the stress testing, which is known as photostability testing, which is an integral part of stress testing, should be conducted on at least one primary batch of finished pharmaceutical product if appropriate. And then how do we select the batches? For finished pharmaceutical products containing new APIs, data from stability studies should be provided on at least 3 primary batches of each proposed strength of FPP. 2 of the 3 batches should be at least pilot scale batches, and the third batch can be smaller. The primary batches should be of the same formulation and packaged in the same container closure system as that proposed for marketing. The manufacturing process used for primary batches should simulate that to be applied to production batches and should provide product of the same quality. And then comes the container closure system. The stability testing should be conducted on the dosage form packaged in the primary container closure system proposed for marketing. If the secondary container closure system has protective properties and labeling clearly indicates that the product is to be stored in the primary and secondary packaging, or if the product is packaged in a semi-permeable container where components from the secondary packaging can migrate into the product, the secondary packaging may also form part of the packaging system for stability samples. The next comes the specifications. The testing should cover as appropriate the physical, chemical, biological, and microbiological attributes, preservative content, and the shelf life acceptance criteria should be derived from consideration of all available stability information. Any differences between the release and shelf life acceptance criteria for antimicrobial preservative content should be supported by a validated correlation of chemical content and preservative effectiveness demonstrated during development of pharmaceutical product within the product in its final formulation intended for marketing. And next comes the testing frequency. For products with a proposed shelf life of at least 12 months, the frequency of testing at the long-term storage condition should normally be every 3 months over the first year, every 6 months over the second year, and annually thereafter throughout the proposed shelf life. The initial date of storage should be considered T0 and stability time points should be defined as a date with respect to T0. For each time point, samples should be withdrawn and tested as per the protocol. Testing should be completed as soon as possible and deviations from the protocol should be recorded and justified. And then comes the storage condition. In general, an FPP should be evaluated under storage conditions with specified tolerances that test its thermal stability and, if applicable, its sensitivity to moisture or potential for solvent loss. The storage conditions and length of studies chosen should be sufficient to cover storage, shipment, and subsequent use with a due regard to the climatic conditions and in which the product is intended to be marketed. And for the general case, if the study is long-term study, the storage condition should be 25 degrees Celsius plus or minus 2 degrees Celsius, 60% RH, or else it should be 30 degrees Celsius and 65% RH, or then it should be 30 degrees Celsius and 75% RH. And the minimum time period covered by data at submission for long-term is 12 months or 6 months. And for the intermediate study, the storage condition is 30 degrees Celsius plus or minus 2 degrees Celsius and 65% RH plus or minus 5% RH. And the minimum time period covered by data is 6 months. And for the accelerated studies, the storage condition is 40 degrees Celsius and 75% relative humidity. And the minimum time period covered by data is 6 months. And for the finished pharmaceutical products packaged in impermeable container, the parameters required to classify the packaging materials as permeable or impermeable depend on the characteristics of the packaging material, such as sealing, thickness, and permeability coefficient. The sensitivity to moisture or potential for solvent loss is not a concern for FPPs packaged in impermeable containers that provide a permanent barrier to passage of moisture or solvent. Thus, stability studies for products stored in impermeable containers can be conducted under any control or ambient RH condition. And then comes the FPPs packaged in semi-permeable containers. Here, for the long-term study, the storage condition is 25 degrees Celsius, 40% RH, and 30 degrees Celsius and 35% RH, and the minimum time period covered by data is 12 months or 6 months. And for intermediate studies, the storage condition is 30 degrees Celsius and 35% RH, and the minimum time period covered is 6 months. And for accelerated study, the storage condition is 40 degrees Celsius and not more than 25% RH, and the minimum time period covered is 6 months. And for the FPPs intended for storage in refrigerated, for the long-term study, the storage condition is 5 degrees Celsius plus or minus 3 degrees Celsius, and the minimum time period covered is 12 months or 6 months. And for the accelerated study, the storage condition is 25 degrees Celsius 60% RH, or 30 degrees Celsius 65% RH, or 30 degrees Celsius 75% RH, and the minimum time period covered by data is 6 months. And next over to Prathamesh.
Good afternoon, ma'am. Let's continue. I continue with the statements and labeling. All the FPP should be packed in a containers that ensure stability and protect the FPP from deterioration. Additional labeling statements should be provided, could be used in case where the results of the stability testing demonstrate some limiting factors. And involved use, in use and hold time stability. The physical and chemical microbial properties of the FPP that are susceptible to change during the storage should be determined over the period of proposed in-use shelf life. If possible, the testing should be performed at an intermediate time points and at the end of the proposed in-use shelf life on the final amount of the final product remaining in the container. Variations. The scope and the design of the stability studies for variation are based upon the knowledge and experience acquired by the APIs and the FPPs. The results of these stability studies should be communicated to the regulatory authorities concerned following the applicable requirements stipulated in the variation guidelines for the region. Ongoing stability studies. The protocol for the ongoing stability program can be different from that one of the initial long-term stability studies as submitted in the marketing authorization dossier, provided that this is justified and documented in the protocol. For example, the frequency of testing as above, like the number of batches or per strength on different batch sizes if applicable, like the relevant physical, chemical, microbiological and biological test parameters with acceptance criteria, like that like the reference to test methods and the description of the container closure system. All these are considered as a summary of all the data generated, including any interim conclusions of the program should be written and maintained. This summary should be subject to the periodic review. That's all from team 3, ma'am.
I hope you understood that stability is not just about just keeping the medicine for stability. It is much beyond it. And as I said, the stability protocol that will be prepared will be very, very elaborate. I can't show you obviously because that's a proprietary information of any company. But companies based on these standards now, for example, since you are already trained now, you should be able to write an SOP on how to do a stability of a sterile finished product. You should be able to write based on these standards. That's what we do generally. That's how we learned how to write an SOP. So there will be an SOP on preparing a stability or conducting a stability program. How to conduct a stability program, there'll be an SOP on it. And based on that, there will be a what do you call template for preparing a stability protocol. What are the components to be addressed? What you just saw now. Again, this is a discussion that happens with the marketing team, the QA, R&D and all that, and then they take a call. And again, different dosage forms or whether there's a bracketing required, whether there is a matrixing required, everything is discussed over there. And then they take a call and decide on the stability study. And once the stability study is sort of fixed, the protocol is derived. And once the protocol, as I said, it's got an SOP, they have their template. And again, the observation data sheets and or the method itself is written over there. And then, of course, they prepare the report. Now, interestingly, over a period of years, when you realize, when you're talking of huge companies, like for example, Syngene, which has a stability vertical, that means thousands of samples gets analyzed. There are teams who does only that. Unlike in other companies where the QC will have one team that handles the stability of the sample, in case of Syngene, because it itself is a vertical, they will have an entire team working only on stability of the samples. That means they will liaise with the client, they'll prepare the protocol, the client will approve it, then they conduct the study. Now, coming to the sample management. If you remember, I said previously in my session that in a lab, there are pre-analytics, analytics, and post-analytics activity. So in case of stability, what is the pre-analytical activity? Now, the sample will come again. In the case of Syngene, because it's a contract research and manufacturing services company, the client will send the sample bottles. That means those bottles or blister packs or whatever it is sent by the client, okay, to Syngene. And what Syngene accession team, sample accession team, the receiving team should do is, once they receive it, of course, you just can't receive saying that, oh, I need so many of this particular drug. There is a import license the regulatory team has to take, and then after it is taken, the shipment has to come. Again, once the shipment has to come, again, some formality will be there. Once it comes into the premises, each and every bottle is verified. Each and every bottle is checked whether it is, all fine, the labeling is fine, the bottle, what do you call, is not opened or tampered with, the seal is not broken. All that will be verified by the team, and then only they will accept it into the system. Now, in many companies, what they have is they have a separate stability program, software program itself that they have generated, they have created. What it does is it will indicate for this program, how many samples will be there, what are the tests to be done, everything will be mapped related to the sample. Just like how QC details are mapped, what tests to be done, etc., like that, in stability also, everything will be mapped. Now, once it is mapped, the labels are generated. If there are hundreds of samples, then 100 labels will be generated. Each will indicate which month it has to be withdrawn for testing. So all those labelings, everything is done, and then it is stored in that particular temperature and RH in the stability chamber. Now, in the system, everything is populated. What you just showed now. Everything is populated. How often should it be retrieved for testing, everything. So what happens is the dates are fixed. Once the dates are fixed, alert message will go to the analyst. If I'm the analyst, I will get an alert message like 3 days. Again, we can customize it. 3 days from now that the sample is there for analysis. So I have to be mentally prepared and also verify whether those reagents for the testing, everything is available. So, and once that is done, then of course the result is all interfaced, it gets populated, and then it helps in creating a tabulated data, and then of course we have to create the report. So this is how company have used this calibration calendar, stability sample analysis calendar in the computerized system. Okay, so it's all, I would say, computerized. Same thing happens for calibration. Same thing happens for equipment maintenance, preventive maintenance. So likewise, companies are using this so that we do not forget to pull out a sample. Because if I, as an analyst, have to do so many sample analysis every day, I don't want to miss out on anything. So I would rather use a alert mechanism. It comes in mail or it comes on phone. So this is how companies execute their stability program. Okay, I take leave and I see you tomorrow. Yeah.
Thank you.
Thank you very much.
Thank you,
Thank you,
ma'am.
ma'am.
Thank you, ma'am.
Can we leave?